Good morning, and thank you for joining us this morning for Winship Grand Rounds. If you are an Emory University or healthcare employee and would like to receive CME credit hours for attending today, the login information can be found in the chat feature on the bottom of your screen. If you have any issues with this webinar or the CME login, please send Julie Hawkins an email or drop a note via the chat feature. This morning, we are pleased to welcome Dr. Greg Lesinski. Dr. Lesinski is an associate professor in the Department of Hematology and Medical Oncology here at Emory University School of Medicine. He will be full professor in September of this year. He received his PhD in biomedical sciences from the Medical College of Ohio in Toledo, Ohio. He obtained his MPH in clinical investigation and completed his postdoctoral training at The Ohio State University in Columbus, Ohio. Dr. Lesinski's research is focused on gaining a greater understanding of the interaction between the host immune system and tumor cells. The ultimate goal is to use the knowledge to develop novel therapeutic and chemopreventive approaches to help patients with cancer and improve upon existing therapies. Of particular interests are modulation of cytokines and downstream signaling pathways to maximize the effect of immune-based therapies. Dr. Lesinski is in addition to his very active research program, he also is the vice chair for basic research in the Department of Hematology and Medical Oncology. He holds a number of professional memberships with AACR and the Society of Immunotherapy of Cancer. Please welcome Dr. Lesinski.
Thank you, Basil, for the introduction, and to Winship for the opportunity to present some of our work related to translational research in pancreatic cancer today. And really, what I hope to convey to you is the fact that we have a very collaborative program. This is something that is not unique to one lab or even one department. And I hope to provide a few examples of true translational research and team science in this disease. So I have a few disclosures related to off-label use of some drugs that are in clinical trials, some consulting, and then research funding that we receive to conduct correlative studies for human clinical trials. So although today's topic is on pancreatic cancer, we also have the disclosure that we will be unable to discuss our work in cholangiocarcinoma, and maybe we can do that next time. But we are very fortunate here at our institution to have a robust translational research program that's focused on pancreatic cancer. This is a program co-led by my colleague Basil and myself, and our overall programmatic goal is to integrate findings from the laboratory into clinical trials for patients. And this is definitely a team effort. We have a number of very active colleagues who are on the clinical side, as well as those in the laboratory side. Many people come to us from multiple departments in both hematology, medical oncology, surg-onc, pathology, radiation oncology, and many others. And on the laboratory side, in addition to our group that is focused primarily on GI oncology and immunotherapy research, sort of one of the things we're excited about is that we've been able to entice other people that may not work regularly in this space to become interested in pancreatic cancer and other GI research. So we really have a great team, and everyone listed in this picture and many who are not contribute to that effort that we're really driving forward here in Atlanta. So just by way of background, so we can all orient this early in the morning for those who are catching up on their coffee consumption, I'm just going to provide a general background and sort of a historical perspective on the role of cancer-associated fibroblasts in the stroma of pancreatic cancer, so we can set the stage and understand where we've been and where we're going. Second, I'll talk a little bit about some emerging data on how these CAFs interact with the pancreatic cancer cells themselves. Third, we'll get into more targeted therapeutics stories, looking at how cytokines derived from these fibroblasts in pancreatic cancer can be used as targets to enhance immunotherapy in this disease. And then finally, I'll end with a few short stories about novel targets that we have funding for to pursue in the fibroblasts to hopefully make immunotherapy work better in the setting of pancreatic cancer. So this disease, pancreatic cancer, is no secret. It is a very aggressive type of tumor, the fourth leading cause of cancer death in America currently. The survival rate over 5 years, as we know, has gone up to 10%, which is, we still have a tremendous amount of work to do. And then some alarming statistics published recently indicate that PDAC will actually surpass both breast and colon cancer and is predicted to become the second leading cause of cancer-related deaths in the coming years. So this is increasing in incidence, and obviously the severity is one that we cannot take lightly. Further complicating matters is the fact that treatment options are not very good. Outside of surgery, when the disease is detected early, which is really the best chance for cure these days, regimens consist primarily of chemotherapy-based. There's some molecular targeted therapies in subsets of patients, and then outside of microsatellite, unstable, or other mutated tumors, immunotherapy has made little impact in this disease. So this last problem, the idea that immunotherapy has not really put a dent in pancreatic cancer survival rates, is a major focus for our laboratory and our larger research group. One of the reasons we believe that might be accounting for this tremendous immune suppression, both in the tumor and systemically in patients, as well as the lack of response to immune therapy, is derived from one histologic feature that's very prominent in pancreatic cancer. And that is essentially the fact that there's a tremendous stromal reaction that occurs in these tumors. So you can see a histologic image here. If you zoom in on a patient's pancreatic tumor, this is brown staining for a fibroblast activation marker called alpha smooth muscle actin. The staining is brown, it surrounds these tumors, it intercalates within the tumors, and these positive cells that we're staining for lay down dense matrices of collagens and other extracellular proteins that render these tumors highly desmoplastic and rock hard if you take them out of a OR in the clinical specimen. So these fibroblasts in essence are, thought to present a physical barrier in many studies, as well as a biological barrier that we'll talk about later throughout this lecture. So kind of an overview, if you look at what types of cells are in the pancreatic tumor microenvironment, to set the stage for research, we certainly know, in addition to the tumors, there are these fibroblasts and other stromal cells that form this complex network. There are certainly immune cells present in the tumor. There seems to be a myeloid cell predominance in these tumors, and much of this process is driven by soluble factors, cytokines, chemokines, other metabolic mediators that are derived from these tumor stromal complexes of cells in the pancreatic tumors. And the presence of fibroblasts is not just in the primary tumors. They've been detected in metastases, which are the leading reason for such rapid decline in patients with pancreatic cancer. And the job is really to identify ways to get T cells either exogenously administered or ones endogenously present T cells to better recognize and eliminate these tumors. So there are a number of forces that prevent this from happening, one of which are indeed soluble factors from the tumors in stroma. Others are the expansion of these suppressive myeloid and other immune type cells in the tumor, and these provide prime opportunity for a number of therapeutic targets. So this is much of the work that's going on here. There's certainly examples, pharmacologic targeting of both the tumor cells and the stroma. We'll talk a bit about that today. We have a very active program of immunotherapy in pancreatic cancer that is gaining momentum every day. There's a number of cell therapy approaches being studied here at Winship by folks like Ned Waller, Crystal Paulos, and Sarish Rafiq in our department. Ourself and other groups are interested in immune checkpoint blockade, both traditional checkpoint blockades and novel checkpoints, such as VIP that Ned's lab studies. There's a number of examples in clinical trials and preclinical work giving agents that will target this myeloid and macrophage component, a trial led by Christina Wu in the GI space targeting semaphorin 4D, for example. Cytokine targeting is a major interest of our lab, and we'll talk more about that today in our work with interleukin-6. And then finally, there's interest in early detection of cancer. Our colleagues at Georgia State, such as Jenny Yang and Ziren Liu, who are developing novel imaging tracers to detect liver mets early, and then work out of Lily Yang and others' laboratory are developing really excellent nanotechnology approaches to get in and target and deliver drugs to the tumor microenvironment. So we're very fortunate to have such a robust program of research here, and excited for all the discoveries to come. So the overall hypothesis of our talk today, and what I hope to address, is the fact that these fibroblasts in pancreatic cancer may harbor key targets that we can leverage therapeutically. And in particular, we can do so to enhance the efficacy of immunotherapy in these patients. One second. So before we can get into the new research, it's always helpful to take a look into history and see what has been done previously. And one of the earliest examples of targeting the stroma in pancreatic cancer was related to a pathway called Sonic Hedgehog, and not just the video game for those who grew up in the era that I did. But the rationale behind this target, or targeting this pathway, was that there were several bits of data indicating that paracrine signaling from this neoplastic cells in the pancreas, from the cancer cells to the stroma, would facilitate this fibrotic reaction that we talked about. And there's an inhibitor of this pathway, smoothened, that will disrupt it and improve the vasculature, and it was thought that it would give greater access to chemotherapy. So Ken Olive's lab at Columbia did a lot of the sentinel work together with Dave Tuvison, some of the titans in the field, and they have shown that, early on, V is for vehicle, G is for gemcitabine, the chemotherapy, and I is for the smoothened inhibitor. If you look at the far left graph here, these are cells undergoing apoptosis, so cleaved caspase-3 positive are certainly greater in mice receiving this drug in combination with chemotherapy. And they also saw in the KPC model, this is a mouse with spontaneously arising pancreatic cancer, a significant but somewhat modest survival benefit in animals receiving both drugs. And notably, these mice also had fewer liver metastases. So this really ignited some excitement in the field. It was stagnant, and now we have a way to target the stroma and go after things, or so we thought anyway. So until things went to clinical trials, and that's where the field really became very confused. And what happened, long story short, was that the clinical trial combining this Hedgehog inhibitor together with gemcitabine, the patients on the placebo arm, unfortunately, did better than those undergoing treatment. So the trial was stopped. And this was a major blow, and launched a number of research initiatives to try to figure out why the preclinical data and the clinical results were so discordant. So upon looking at the data more closely, they found that the duration of smoothened inhibition was quite short in the animal study, really around 3 weeks. And work from this same group and others, including a paper that we were involved in 2018, indicated subsequent studies that smoothened inhibition would act on multiple cellular compartments, including the fibroblasts. And this by itself would facilitate more aggressive tumors and alter this cellular crosstalk that indeed occurs between the tumors and the stroma. At the same time, there was another ongoing study with a different drug targeting similar aspects of the pathway, combined with gemcitabine again. And unfortunately, this trial consistently also showed no therapeutic benefit. So work from Andy Rim's lab, again involving Ken Olive and Ben Stanger and others, indicated that if you would ablate this pathway in the cancer cells themselves, what you see here is a Kaplan-Meier curve of the mice. They have spontaneously arising pancreatic cancer, and they've lost Sonic Hedgehog, a component of this pathway. they progress much faster in their cancer and have a poor overall survival. So these really sophisticated conditional knockout mouse studies in this genetic model of pancreatic cancer then lended further insight to why perhaps this trial was unsuccessful and identified a role for the pancreatic stroma, not in promoting tumors, but restraining their progression. So this really ignited some controversy in the field and said to us, you know, we really have to learn more. We really have to be careful if we're going to target the stroma or components of the stroma and study them very vigorously before launching into clinical trials. So, you know, historically, again, sort of the last bits of background here, we asked the question of what are the consequences of eliminating CAF? So a number of sentinel studies from various groups used sophisticated genetic approaches to do this. The first was from Rahul Kaluri's lab in Cancer Cell 2014. This was published together with the last paper from Andy Rim's lab, showing that when you selectively deplete these alpha SMA positive fibroblasts from mice, the cancers progress faster. These again are mice with spontaneously arising pancreatic cancer. So this again agreed with the data and said, if you get rid of, you know, a chunk of these stromal cells altogether, the cancers go faster. They become more aggressive, more metastatic. And then the same group looked in a retrospective cohort of patient tissues and showed very similar trends. The red line are patients stratified into their primary tumors having lower expression of this activated fibroblast marker, smooth muscle actin. And these patients had a shorter overall survival that was significant when compared to tumors expressing high SMA. Again, arguing that the stroma was protective to patient survival. Now, with that said, there's always multiple sides to every coin when it comes to science, and creating further confusion, but I would also argue great opportunity, was a series of studies from Doug Fearon's group showing that these fibroblasts, different subsets this time, FAP positive subsets, which are heterogeneous themselves, they can secrete chemokines like CXCL12 that lead to immune exclusion. And then using both genetic approaches as well as pharmacologic approaches by targeting the receptor, CXCR4, they showed that tumors, if you modulate these fibroblast compartments, can be made more sensitive to immunotherapy approaches like PD-L1 or CTLA-4. So notably in the prior study, they showed similar phenomenons in that the alpha SMA depleted mice were more sensitive to anti-CTLA-4 blockade. So taken together, these data might suggest, although the stroma is holding the tumors in check, it may also be a target to sensitize to immunotherapy. So this is definitely something that interested us. And then our colleague Dave Tuvison, who gave grand rounds a few months ago, has really identified further aspects of fibroblasts that are of interest, and notably their heterogeneity. So Dr. Tuvison's group has used a variety of single cell approaches to refine our understanding and subclassify fibroblasts into different subsets in the setting of pancreatic cancer. So the first are these alpha SMA positive fibroblasts we've been talking a lot about. They're more myofibroblasts-like. Their job is to lay down collagen and promote this desmoplasia. They're typically found proximal to the actual tumor cells in the tumors. And then, you know, in contrast to that, there are other subsets, the inflammatory type of CAFs, that are more distal from the tumor lesions, more proximal to the immune cells that are excluded. And these subsets of fibroblasts might secrete various chemokines and cytokines like IL-6 that facilitate this immune profile of the tumors and alter the landscape. And then finally, a more less frequent subset, the antigen-presenting CAFs or AP CAFs express a molecule called CD74 and might present antigens to CD4 T cells. So this area is moving really, really rapidly. There's been an explosion of publications on single cell RNA-seq that's identifying even more targets from various groups that might be relevant to the biology of these fibroblasts in pancreatic cancer. So we have a lot of work to do, but one limitation, you know, if you're writing a grant, you want to talk about the rigor of prior research, for example, is the fact that most of the data on these fibroblasts thus far aside from a few select studies, have been really limited to primary tumors, and we don't necessarily have a comprehensive understanding of what their role is in pancreatic cancer metastases, and certainly not a complete understanding of how they communicate with the immune system. So Mara Sherman published a really nice review article, also emphasizing the fact of, there's a tremendous amount of heterogeneity. Everyone has their favorite marker, but above all, this process is really heavily regulated and influenced by the different cytokines that are present in pancreatic tumor microenvironments. So this is an opportunity for targeting cytokines to modulate the stroma. And, obviously there's a lot of markers here, so this has provided a tremendous amount of confusion and uncertainty to the field. What ones do we look at? What are the right markers? What are the wrong markers? How do we even keep up with this? And I think Twitter did a good job of distilling that down very simply. And, taking this tongue in cheek, the first article here in Types of Pancreatic Cancer Papers is one our laboratory is certainly guilty of. Stellate cells, or CAFs, they might do things in the pancreas. They do things to the cancer, they do things to the immune system. And we'll certainly talk more about this, but it's an active area investigation. The second is the explosion of single-cell RNA-seq to identify individual fibroblast subsets. First, it was 2 fibroblast populations. Then in order to get a cancer discovery paper, you had to get 4 fibroblast populations, which has been done. And now the bar has been set much higher. In order to get into a high impact journal, you've got to identify over 25 fibroblast populations. So this is, a very cheeky summary of the state of the field, but hopefully we'll continue to learn more. So what are we doing in our lab and in our group here? We're really looking, not so much at defining new subsets of fibroblasts, sort of like everybody's doing that now, right? But what we are trying to do is identify functional relationships between the stroma, these CAFs, in the pancreatic cancer cell, as well as what they may be making or communicating to the immune system with, and how can we then throw a wrench in that and intervene to make immunotherapy better. So this is sort of what we're doing and what I'll spend the rest of the time talking about today. And one of the things that we've been able to do pretty well and is sort of an interesting direction is take human pancreatic cancer cells, develop them into 3-dimensional cultures as spheroids, and then ask the question, what happens if we take cell culture supernatants from patient-derived CAFs of pancreatic cancer and just simply put them on the cancer? You can look at them through microscopy using a variety of different metrics to describe the shape of the cells or invasive phenotypes and other features. And my former graduate student, Brandon Ware, this was really his conception of doing this project. And some of the still microscopy images that he has generated initially is when you take these collagen matrix embedded pancreatic cancer cells that are human, expose them to CAF supernatants from a few patients, and then look at the phenotype of the cells in culture. You can see they assume a sheet-like invasive phenotype. They spread out when just cells alone exposed to normal media do not do that. So there's definitely something going on here that's of interest and scientifically, piquing our curiosity. And Brandon took a video of this in collaboration with our awesome integrated cell imaging core. You can see the cells spread out, exposed to supernatants. If you label the nucleus in blue, if you follow this cell down at the bottom, you can see there are several very metabolically hyperactive cells that emanate from this tumor mass. And they sort of survey the periphery of the spheroid as it spreads out and grows. And we sort of identified these cells akin to sheepdogs that were herding the flock. And we don't really know whether these are good or bad or indifferent. What they're doing, are they laying down different types of collagen? Are they restraining? Are they guiding? This is an area that we're very interested in studying. And if you look at the still image of this video over here, you can see them sort of aligned along the invasive edge of the spheroid. And this is really interesting to us. And we were fortunate to get Winship Invest pilot funding. This is a collaboration with the Marcus Lab. And what we've done in collaboration with Carol, Jana, and Joseph from Adam's lab. My graduate student, Emily Green, is leading this on our end. We have put the Dendra 2 construct that's photoconvertible into the pancreatic cancer cells. We can shine a laser on them to actually turn the cells from green to red and then isolate only the ones we want. So we're undergoing studies where we take out these sheepdog cells as well as cells from the larger pack and sort them by flow, and then do RNA-seq to ask several factors. First being, what are the dominant gene expression profiles induced from soluble factors derived from the CAFs? And might that identify pathways that we can target to limit this cellular behavior and potentially the invasive phenotype of the pancreatic cancer cells? And this work was really inspired largely out of Adam's groundbreaking work from his group, through Emily and Rachel and others that came before that did this saga spatiogenomic and cellular analysis on lung cancer cells and identified leader and follower cells. So again, this is a fabulous example of getting people interested who normally don't do pancreatic cancer work into the field. So to shift gears a little bit, in addition to the ability of pancreatic CAFs to communicate with the cancer, we know that they can certainly communicate with the immune system, and this is absolutely an opportunity for improving therapies in this disease. My former postdoc, Tom Mace, this work goes all the way back to 2013, where Tom collaborated very well with our surgical oncologists at Ohio State at the time, and now this work is carried on with Drs. Mythel and Sarmiento here at Emory, where we can get pancreatic tumor surgery specimens, isolate out the stellate cells or the CAFs, and Tom identified a number of soluble factors using a bioplex assay in the supernatants of these cells. And one of the things that emerged, if you look at 7 individual patient-derived CAFs compared to a normal pancreatic fibroblast, was that interleukin-6 was remarkably upregulated and secreted from these cells. So this was very interesting to us. And Tom later went on to show and publish in Cancer Research that these supernatants, if you'd put them on normal immune cells, you could drive the differentiation of suppressive functional myeloid cells. They could inhibit T cell proliferation in vitro of autologous T cells. And at the mechanistic level, he showed that this process occurred in a manner that was dependent on this IL-6, and it was dependent on downstream STAT3 signaling in the CAFs. So this implied immune crosstalk might be possible from the things that are secreted by these pancreatic cancer-derived CAFs. So fast forward a few years, we decided to take a little more unsupervised approach, doing sort of the same thing where you get pancreatic tumors, you isolate the CAFs out, and now we did a nanostring profile on the RNA that resulted from them. And you get these heat maps that look very pretty, where red is upregulated gene expression, green is downregulated. And here we have disease or CAFs derived from 10 separate primary pancreatic resections or 2 normal fibroblasts. And one of the interesting things that emerged in agreement with our protein data was the fact that IL-6 was the most abundantly expressed cytokine transcript in all of these cancer-associated fibroblasts. So we also were fortunate to identify several new targets that we're systematically investigating in the laboratory and may or may not be good, but that's the topic of future research for us for sure. So for those who don't live in the IL-6 world, this is a really cool cytokine. It will bind its receptor that can be both membrane bound or soluble, allowing this cytokine to signal in trans to cells that don't have the receptor. And they do so by transducing signals through this other protein called GP130. That's a transmembrane protein. It's the anchor for a lot of the signaling pathways. And when IL-6 and its receptor complex engage GP130, either classical or in trans, it will facilitate the activation through a series of phosphorylation events of a number of inflammatory pathways like JAK-STAT, MAP kinase, NF-kappa B, and others. So this pathway is involved in immune suppression, of course. It can come from a number of sources. Our work has focused on the CAF components of pancreatic tumors, but cancer cells themselves in certain cases can make IL-6, and so can myeloid cells. So there's a lot of built-in redundancy in that this cytokine can signal through several cells and come from multiple cells. So in essence, it may be really instrumental to the biology and the immune phenotype of these tumors. We know that it can bind to these receptors and have all the bad things happen on cancer cells that you don't want. It can facilitate increased proliferation and progression, driving metastatic gene expression, maintain stem-like cells in the setting of the tumor cell. And then with regard to immune modulatory properties, IL-6 is known to facilitate expansion of MDSCs as well as different subsets of T cells like Th17 or other macrophages that may be sort of keeping this inflammatory phenotype of pancreatic tumors alive and well. So this led us to a model by which we thought IL-6 and other factors were acting and identify potential therapeutic targets that we could then pursue. So we know that the tumor stroma and the pancreatic cancer cells themselves maintain a nice reciprocal relationship where they will facilitate one another's proliferation and progression. There are certainly intrinsic cytokine circuits through various signaling pathways within the CAFs and other components of the tumor stroma themselves. We know that these tumors, when they get more advanced, they can secrete immunomodulatory factors at the systemic level, driving upregulation of IL-6 and other myeloid or sort of tropic chemokines and cytokine mediators. And then these factors can act directly on cytotoxic cells that might be trying to make an attempt to eliminate and control these tumors, but they can also, through the activation of many of these same inflammatory pathways, foster the expansion of suppressive myeloid cells that will feed back and provide another layer of immune suppression against the T cells that are trying to eliminate these tumors. So if you see the red boxes on here, these were the nodes that we initially tried to target. We went after cytokines that are highly stromal derived like IL-6 and concurrently attempted to get the immune checkpoint blockade going with things like PD-1 and CTLA-4. So before doing that, Tom had looked in histologic specimens from patient tumors with pancreatic cancer and confirmed there is indeed elevated levels of IL-6 staining in the tumor microenvironment. So this is a phenomenon relevant to patients, not just cells in culture or mice. And then my other former postdoc, Matt Farron, had access to a really nice cohort of plasma samples from patients with metastatic pancreatic cancer who had not been pretreated. So these were, I think, a little over 70 patients. And if you stratify the data based on circulating IL-6 being high or low, the red line in this Kaplan-Meier curve indicates that the patients with higher levels of IL-6 above the median did significantly worse in terms of overall survival compared to those patients who had lower IL-6. So this indicates that the IL-6 is relevant both in the tumor microenvironment as well as the systemic circulation, and again, lends further to support to this potentially being a target for therapy in this disease. So we went back to the mouse where we have immune competent, otherwise normal mice that we can inject a syngeneic pancreatic tumor into, let it grow, and then treat with antibodies that might target IL-6, PD-L1, or both agents combined. And we actually did this study in 4 separate immune competent pancreatic cancer models. I'm just showing you a little bit of those examples today as a lot of this work's published. And what happened, long story short, in one of these models, we're looking at tumor volume, so tumor growth over time, and the orange bar is the rate of tumor growth in mice with established tumors receiving dual antibody blockade against IL-6 and PD-L1. You can see these mice, the tumors progress much slower, significantly slower than either single agents or our controls. And notably, blocking IL-6 alone does very little in this model. But when we look in the TILs of these tumors taken at the study endpoint, we see that there is an increase in CD8 T cells that are of an effector phenotype. So targeting these pathways would actually invigorate T cell infiltration into these tumors and hold them at bay much longer. And other mechanistic studies show that this whole process was indeed dependent on the CD8 T cell compartment. If you deplete those cells, the effect goes away. So just a little more representative in vivo data from another model. This work was done in collaboration with our colleagues at Ohio State, Rina Shakya, Thomas Ludwig, and Micah Strowski, who's now at MUSC. And they have a really cool animal model whereby this is a spontaneously arising pancreatic cancer on the KPC background. So a KRAS P53 mutant-driven cancer with an additional mutation in BRCA2. And what results are mice that get pancreatic cancer with 100% penetrance at 5 weeks of age. So this is really good for therapeutic studies in a GEMM model in that they arise rapidly, you don't have to pay for them, and they recapitulate the biology of at least a subset of pancreatic cancer patients that are out there. And what you can see histologically looking after a short course of treatment in these animals, the controls have poorly differentiated tumors. Single agents, there's, the adenos are there, but maybe a little less aggressive. And then in the combination treated mice, we see that the tumors are definitely less aggressive, and even there are some normal acinar structures visible in the pancreas via histologic analysis. So the organ itself with these continually arising spontaneous tumors maintains some of its normal morphology. And we worked with our pathologist at the time, Ben Swanson, to score all these tumors from the animals. And he did a pathologic scoring system whereby he graded low-grade PANNs, or either the high-grade PANNs or frank adenocarcinoma as blue in the graph. And you can see that with more treatments, the dual blockade, there's less blue. And when you deplete CD8 T cells, this effect goes away, rendering the tumors again more aggressive. So looking histologically at these same tumors, what happens to the immune cells? We did some of that work, and this graph down here from the combination treated mice shows more brown spots that correspond to more T cells. And if you quantitate that, there were certainly more CD3 positive T cells in the tumors from the combination treated mice, again, agreeing with the data that we had from the other animal models. So this definitely implied an immune modulatory effect was occurring from this treatment combination. So we were fortunate to publish this work in Gut. It represented a kind of an odyssey of collaborating between lab and clinic. And importantly, our work was followed by several other papers and consistent across multiple tumor models. There was a paper showing IL-6 PD-1 pathway blockade was efficacious in melanoma, in glioblastoma, and later in colorectal cancers. So it's always nice when other people's work agrees with yours, and the mechanisms were relatively similar across these different tumor models. So this effect was definitely something that caught our eye. And finally, we have R01 funding now to dig a little more into the mechanism of how this works. And we're working with Micah Strowski at MUSC, who's a really great mouse modeler and has developed a very sophisticated dual promoter system. And I apologize to the clinicians this early in the morning for the very heavy basic science on this slide, but this mouse is really cool in that they can drive expression of oncogenic KRAS and delete out P53 with a recombinase in the pancreatic epithelial cells, while at the same time using a different promoter system, excise IL-6 from cells of the fibroblast lineage. So what you get is a conditional knockout mouse with pancreatic cancer that arises naturally in the organ, while at the same time you lack IL-6 in these fibroblasts that we think are a major source of it. And when we ended the study at day 90 in several of these mice, this is early on data, we see that the PDAC lesions graded by histology are much less in the conditional knockout mice. And the tumor weight in these animals at this time point is also much lower, again, agreeing with the histology. So together, along with our data, these data are really providing additional mechanism to show that IL-6, specifically in the stromal CAFs, is important for the progression of pancreatic tumors. So the thing we're most excited about is now the ability to take this 7+ years of work from 2013 to 2020 from the lab all the way into the clinic, where now we've been able and fortunate to translate these findings to a human clinical trial. And this work was really driven through the energy of Dr. El Reyes, who we collaborated closely with our pharmaceutical partners to do a phase 1b/2 clinical trial of the IL-6 blocking antibody siltuximab and spartalizumab. The dose expansion cohort has been completed in the patients, or dose escalation cohort, I'm sorry. And we're now in the expansion cohort where patients will enroll in the trial. They'll get a pre-treatment biopsy. They'll get a single treatment with either PD-1 or the combination, followed by another biopsy, usually of the liver in metastatic disease. And then everyone crosses over to go on spartalizumab and siltuximab. So we're getting both tissue and peripheral blood to really dig into the correlates. And a lot of that work is really being spearheaded by my current postdoc, Cameron Herding. So what are we going to do with all this tissue? Basil and I just received news a few weeks ago of a really favorably scored R21 grant. We got a 2 percentile. So we were literally jumping up and down at the opportunity to dig in deeper to these rare tumor samples and ask a number of key questions. The first being this idea of fibroblast heterogeneity. And fibroblast heterogeneity, not in the primary tumor where we know so much, but in metastatic tumors, in liver mets from these patients. So we're actually interrogating the differential expression of various markers related to the MyCAF, ICAF, and APCAF subsets. And also, it's important for us to dig into the immune mechanisms in the tumor microenvironment, as well as systemic circulation as well, as part of this proposal. And our key questions are, first, to determine whether or not we see a similar degree of T-cell infiltration in patients, as we did in multiple murine models. Second, we want to ask, what is the actual nature of these T-cells, given that IL-6 can regulate expansion of different T-cell subsets like Tregs, Th17. Certainly being at Emory, in collaboration with Dr. Ahmed and his group, we want to look at the impact of this therapeutic regimen on different memory T-cell subsets, including the TCF1 positive stem-like T-cells. And then we want to follow with these clinical specimens some important work published from Bob Vonderheid's group, indicating that IL-6 in pancreatic tumors actually allows for apoptosis of type 1 dendritic cells to occur, and early on facilitates lack of effective T-cell infiltration into these tumors. So these are very interesting research questions that we're excited to move forth on, and hopefully we'll be able to impact the field. So now, in addition to these idea of targeting cytokines in combination with other immune checkpoints, there's a number of other studies going on, too numerous to talk about here, but we'll touch on a few that are at early stages and we're also very enthusiastic about. And the first is using an agent that targets the cancer cell as well as a number of other compartments. And this is an inhibitor of heat shock protein 90, a known chaperone protein. And this chaperone will bind to client proteins in the cell, regulating their activity. And many of these client proteins of HSP90 are actually those that are instrumental in cell survival, proliferation, metastasis of cancer cells. So we have a collaboration with a company, Exelixis, that they've made a drug called XL888. It binds to the N-terminus of the HSP90 in the ATP pocket, and it prevents the client proteins from binding it and results in their proteasomal degradation. So they go away, the cancer cells die, and hopefully we can use it in combination with other drugs. So Raju from Dr. El Reyes' lab has done a lot of work and published data indicating HSP90 inhibitors have direct anti-tumor effects. There have been some studies showing how they may function as immune modulators. And then finally, the question we wanted to ask is whether or not they would impact the stroma in this disease. So we've been fortunate at Emory to work with a great team on the clinical side in both surge path as well as pathology, folks like Dr. Krasinskis, as well as Dr. Mythel and Sarmiento, where we can obtain patient specimens to isolate CAFs and look at how drugs affect them in the preclinical setting. So this is an example of looking at how HSP90 inhibitors modulate the activation of CAFs. And what you can see, a positive control treating CAFs from patients with retinoic acid will cause them to turn red. And this is because we add labeled lipid droplets that accumulate when the cells are quiescent. So in their normal state and just media, there's no red. They're not accumulating anything. They're kicking out all these cytokines and other things we've shown. But when you put HSP90 inhibitors on the stellate cells, this had never been done before, you see the red sort of accumulate that we've graphically represented here. And if you look by Western blot at a number of these other proteins, alpha smooth muscle actin goes down, implying the cells are less activated. Phospho-STAT3, a pathway involved in HSP90 regulation, also goes down, implying that treatment of these cells has this attenuated phenotype. So we launched a series of in vivo studies. This was led by our visiting medical student, Yushen Zhang, together with our surgery resident, Mohammed Zaidi, and Brandon and Hannah, our former graduate students. We have luciferase expressing pancreatic cancer cells. We can implant using a surgical procedure in the pancreas of immune competent mice. And what happens if you image them, the mice, their pancreas actually glows. So you can look at the tumor burden over time. Pretty cool technology. And then we take the tumor weight at the end of the study and see whether or not the data from BLI, this bioluminescent imaging, agrees. And sure enough, in this study that we published just this year in Molecular Cancer Therapeutics, mice treated with the combination have smaller tumors than those in the other groups. And if we look histologically, this work was done with our cancer tissue pathology core. The alpha SMA in the tumors is lower in the combination, again, agreeing with our in vitro data. And importantly, the CD8 T cells went up. So this is showing that by targeting this activation phenotype in the stellate cells, we may be getting more T cells in the tumor and less activation. So this has also launched into a clinical trial. And this work is funded through another R01 that's MPI between myself and Basil. And the clinical trial is open to both pancreatic cancer and colorectal cancer. And again, we have this design where we get a short course of either PD-1 or the combination, and both patients, both arms of patients then go on to the combination therapy. So my postdoc, Cameron, is really digging into the analysis of these specimens together with Rafi and of course Basil. And again, this is what the biopsy and blood collection looks like, where everybody gets biopsies pre and post, as well as blood. And then we're doing a number of immune correlates on the plasma, the white blood cells, and then of course the tumor biopsies. So using technology available through the Winship immune monitoring core that was set up by Madhav and Kavita Dodapkar, we've really been guided tremendously by Dion Doxey, who's a fabulous scientist, to set up CyTOF optimization and assay where we can take biopsies from these liver mets, process them to a single cell suspension, and then Cameron has worked with Dion to work out a 37 marker panel to collect data by mass cytometry and really make sense of what's going on both in the blood and in the biopsies. And sort of an early preview of what some of that data is coming out to look like. We can isolate live CD485 positive and negative cells. So these are immune, these are tumors. And then we know that in these liver biopsies from our PDAC patients on trial, this is what the cells look like in the tumors from liver mets. We have a high degree of CD11B positive myeloid cells. There's a subset of patients with high B cells, which was quite interesting. And then as we sort of expected from the prior tumors in the literature, the CD8 T cell infiltrate was a bit lower than the other cell types, but indeed was present. And preliminarily looking at pre and post treatment, these are Tisney plots. So the red is a high signal for these cells. There were indeed changes when the data were concatenated together from this cohort of patients in the T cell compartments, as well as the myeloid cells, where we see this dramatic increase in CD11B positive cells post-treatment. So we're now digging deeper into the identity of each of these cells at the single cell level and correlating that with what the clinical outcomes data are from these patients. And this will be something that we really hopefully make some major progress on over the next few months. So we've talked about IL-6, we've talked about HSP90, but there's all these other potential targets in the pancreatic stroma that might be used to potentially enhance immunotherapy. And in order to try to get some clues into where to go, on our road to success, we went back to our heat map. And one of the things that really caught our eye was this protein at the almost top of the list, right above IL-6. And it's here, it's known as DPP4, also known as CD26. And CD26 is a really interesting protein. It has a number of cellular functions, multiple sites intracellular. It can interact with other molecules. And then the extracellular domain, it has the ability to convert adenosine to inosine. And most notably, it can interact with collagen, which is at high levels in pancreatic cancer. And then, of course, there's an enzymatically active site in this molecule that can cleave off chemokines to modulate their ability to attract immune cells. So the other cool thing about CD26 is that there's an FDA-approved drug called sitagliptin that's used for diabetes. So this sort of offers the potential that we might be able to repurpose something for immunotherapy in the setting of pancreatic cancer that's expressed in a unique subset. So we were also fortunate that very recently Emory recruited Dr. Crystal Paulos from MUSC, and her lab has done a lot of work in studying CD26 and its role in T cells, not in the setting of the tumor cell, but as a way to make the T cells hyperactive in cancer therapy. So my graduate student, Maggie Phillips, validated our gene expression data at the protein level, showing CAFs will express much higher levels of these cells, of this molecule compared to the cancer, although they do have some of it. And then we use PBMCs as our positive control. And then Maggie also validated this at the histologic level by immunofluorescence, showing that stellate cells indeed have abundant expression of the CD26 protein. So in collaboration with Crystal's lab and with Basil on the clinical side, we're really digging into this protein. And our overall model is that CD26 may play an important role in regulating the immune composition of these pancreatic tumors. So we're now taking both pharmacologic with sitagliptin and genetic approaches with CRISPR to delete out this molecule in the cancer-associated fibroblasts and see what happens. So right before COVID and right before we had to shut down this study early, and my student Brandon at the time had graduated, we were able to get a little bit of in vivo data on what happens when you treat mice with these glowing tumors in the pancreas with sitagliptin in combination with anti-PD-L1. And what you see here is a waterfall plot of the bioluminescent imaging. So anything above the line means the tumors grew. Anything below the line means the tumors were shrinking in the combination treated mice. And there was indeed some signals of efficacy in this pilot study. Again, that was unfortunately was cut short by the pandemic. So now we're repeating this, and we're very grateful to the Morningside Foundation for funding this proposal and really digging into the idea as to whether this FDA-approved drug, sitagliptin, may be uniquely suited as an immune modulator in the setting of pancreatic cancer. And this is a highly collaborative effort between the Paulos lab, our lab, and the El Reyes lab. So, you know, just to end things off, I appreciate everyone sticking with me till the end. But what we can conclude from all this work is that CAFs may play a context-dependent role in pancreatic cancer, and we obviously need to dig in deeper into the heterogeneity and plasticity of these cells. There's a number of targets, some of which I've talked about, IL-6, HSP90, and CD26, that may be prime ways to manipulate the immune response in tumors and enhance the efficacy of immunotherapy. And then obviously we have a number of ongoing translational research approaches that we can validate the merit of targeting as a way to disrupt this cellular crosstalk. So thank you for your attention. And, you know, I'll end with the most important slide. This work is really, it takes a tremendous amount of energy and people. All the folks in my own lab, both current and former, that have contributed to this are close collaborators in the El Reyes lab, of course, the Paulos lab, the Marcus lab, and the Waller lab here at Emory. We're really working together to try and make an impact on this disease. Many other colleagues here at Emory that have contributed, as well as other institutions, Georgia State, Georgia Tech, Drs. DiNardo and Tuvison for many critical reagents, Dr. Saab at Mayo for several ongoing clinical collaborations, and then our colleagues at Ohio State and MUSC, as well as our industry partners. And then, most importantly, when doing clinical trials work, you have to acknowledge all patients and their families for having the confidence in science to enroll trials and play a part in making an impact in this disease. So thank you all for your attention, and I'll turn it over to Basil. If there are any questions, I'm happy to address them.
Thanks, Greg, for this excellent grand rounds. If you have any questions, please use the Q&A feature located at the bottom of the screen. While we wait on questions, I wanted to mention that the next week we will hear from our Emory's own Rafiq Pierre Sekali, who will be presenting a grand rounds from HIV to cancer, defining determinants of successful immunotherapy. To view all upcoming Winship Grand Round lectures, please visit Grand Round page on Winship Cancer Center website or the Winship calendar. We already have a few questions, Greg, in the Q&A, so we can go ahead and maybe try and tackle those.
Sure.
First one, can you comment on tumor-associated B lymphocytes? And is there a relationship between tumor-associated B lymphocytes and fibroblasts?
Yeah, that's an excellent question. There's actually quite a bit of work emerging in this space. Some of the Sentinel papers have indicated that B cells producing a cytokine called IL-35 may be facilitating the immune suppression to some degree in this disease. So, as far as their crosstalk with the CAFs, I think that's an area that's, open for opportunity. Certainly the CAFs have abundant cytokine receptors in anything that's made by B cells, perhaps in individual subsets of patients. Some may have more B cells than others that we've shown by our preliminary profiling of the metastatic disease. So I don't think there's anything, immediately known about how exactly they crosstalk. Certainly in addition to the cytokines, the metabolic mediators produced by B cells may be important. And by and large, people in the field are viewing B cells as sort of adversarial to the immune response in the setting of pancreatic cancer. At least that's the data to date that I'm aware of.
Excellent. The next question is from Dr. Lawson. Can you culture the sheepdog cells? And the question he really wants to ask, I guess one question would be whether they act like sheepdogs if there are no sheeps, i.e. no spheroids. Is the spheroid what's making them act this way, or is it something intrinsic to them that's leading that type of activity, I guess?
Yeah, we have a lot of work to do on those cells. And, the first place we're going is the molecular profiling with the SAGA technique. And certainly there's open doors for all sorts of individual cell culture experiments. Again, a lot of this work we're learning a great deal from Adam and his lab and their experience with leader follower cells in lung cancer. And with the ability to photoconvert these cells and isolate them by flow, we can absolutely do those experiments where we take them out and we put them in single cultures to determine whether or not they maintain that phenotype. Do they need the presence of the larger spheroid to maintain it? Or is it solely driven by exposure to the media from the CAFs? I think these are all important questions, and it's just too early to answer your questions, but I'll certainly keep you posted. Thanks for it's a great question.
Okay. And then last question we have is from Dr. Gillespie, who's our new Associate Director for Community Engagement and Outreach. Her question is, fantastic work. Data vary regarding outcomes for Blacks with pancreatic cancer. Do you have info on demographics related to your biospecimens, or any preliminary work that focuses on racial and ethnic disparities in pancreas cancer from your lab?
Yeah, so that's an important point. From our own lab, we do not have that data outside of clinical trial specimens. So from the metastatic data that we showed with IL-6, we have the most access to those data sets. And if I remember correctly, that in multivariable models or univariate analysis, race was not a predictor of survival in this very small cohort. With that said, I think there's abundant literature out there indicating part of the change in incidence of pancreatic cancer, the increase that I talked about very early, it's projected to become a leading cause of cancer-related death, is driven by several factors, one being obesity and the obesity epidemic in the United States. The second is the change in the racial demographics and makeup, where this disease is certainly highly prominent and prevalent in African Americans. So I think these are important areas that we need to address. And I look forward to your leadership, and maybe we can talk offline after this to come up with some brainstorming ideas how we can engage that here at Winship. With regard to the other molecular data that we have, these were typically obtained, like the fibroblast cells, in cohorts of 7 to 10 patients. And most of those specimens, for purposes of the IRB and HIPAA, were de-identified. So we don't have any data on the molecular underpinnings and whether there might be some sort of differences there. But thank you for the question. And I think this is definitely an area that at Winship, given the demographics in the Atlanta area, is a big opportunity for us to dig into a little deeper.
Okay, excellent. I think we're out of time. Thank you, Greg, for a really interesting and remarkable grand rounds. Thank you, everybody.